Howdy. All right, let's continue on with oxygen binding to myoglobin versus hemoglobin. Now, in deoxymyoglobin and in deoxyhemoglobin, the ferrous iron, that iron ball that we saw in the center of the porphyrin ring, actually bulges up above the plane of the heme a little bit. That means when oxygen isn't there as the sixth ligand underneath to pull it down, it actually pooches up towards that F8, that histidine F8 ligand. However, when oxygen binds to the Fe2 in the heme of myoglobin or hemoglobin, the heme iron is drawn toward the plane of the protoporphyrin ring. So it's pulling, the oxygen is actually pulling the iron down into the ring, making that protoporphyrin ring heme with its iron a little bit flatter. So this is only a small change. We go from 0.055 nanometers above the ring, the heme plane, to 0.026 nanometers above the plane of the heme. And so it's not a very big change. And in fact, in myoglobin, which is a single polypeptide chain, that change in the position of the heme iron isn't really consequential at all. But the same change in hemoglobin initiates a series of conformational changes that are transmitted between the subunits. And so it's that quaternary structure of hemoglobin, which is lacking in myoglobin, that makes oxygen binding cooperative and makes the binding curve sigmoidal. In fact, you cannot have cooperativity if you are a monomeric protein. Only proteins with quaternary structure can have cooperativity. And so that's something that harkens back to those, the hierarchy of protein structure. Tertiary structure only, no cooperativity. Quaternary structure, now you can have cooperativity. That's one of the things that's different between myoglobin and hemoglobin, despite their similar structure in terms of their secondary structure and tertiary structure. All right, so that makes sense when you think of the functions of these two proteins. Myoglobin, which is its function is to store oxygen, has a high affinity for oxygen at almost all oxygen pressures. Unless the muscles are under huge demand, say for fight or flight, the myoglobin is going to remain bound to its oxygen. Hemoglobin is different. It has to release the oxygen at the capillary tissue interface. And so it has to be loaded up with oxygen in the lungs at that lung artery interface where the partial pressure of oxygen is high. Remember, that's where it was 100 torr on our graph. And then when it gets to the capillaries at the tissue vein interface, the oxygen concentration is low, about 40 torr, and that's where oxygen needs to be dumped, right? We need to have that catastrophic release of oxygen. And catastrophic maybe is the wrong word choice, but we need to have that all or nothing release of oxygen at the right place. Excuse me. All right, so the binding of oxygen to the hemoglobin is cooperative and its release is also cooperative. What does cooperative mean? It means that it's all or nothing, and it's what gives us that sigmoidal S-shaped curve. This is really well described by Hank Green in his crash course video. So look at that video and pay attention to the cooperativity. Hank makes the analogy of friends sitting around a table or being on a first date. It's very hard for the first oxygen to bind. Remember, we're going to have 4 oxygens ultimately, 1 per subunit of the tetramer. But binding, once you get to high enough oxygen concentrations, you're at the artery lung interface. There's enough high concentration of oxygen molecules to force the binding, to push the oxygen onto one of those subunits. And once one subunit binds to oxygen, that causes that minor conformational change at the heme that actually shoots out an elbow from that subunit to budge the neighboring subunit, which allows it to change conformation and bind to an oxygen and shoot out its elbow to the next neighbor, and the last one binds. So the binding of the first one is hard, but binding of the next three is easy. Okay, that's cooperativity. All right, so this is the hyperbolic oxygen binding curve of myoglobin shown here. And what you can see is that it binds at relatively low concentrations of oxygen or partial pressure of oxygen. Okay, it's a hyperbolic shape, and we'll see this shape, this binding curve shape, again when we talk about enzymes binding to their substrates. It's a hyperbolic saturation curve. Okay, that's the main take-home message from here. Recognize the shape of a myoglobin curve. By contrast, oxygen binding to hemoglobin has a sigmoidal shape or an S shape, and that means that it doesn't bind easily to oxygen until you get to higher concentrations, and then there's a switch over to the R state, which easily binds. Remember I said the T state has a low affinity. T state, T state, T state, R state, R state, R state, right? So everybody's in T in the deoxy state, and then once one oxygen binds to a hemoglobin tetramer, all the other subunits can bind, and that's the hallmark of cooperativity, is this S-shaped or sigmoidal curve. Don't worry too much unless you're studying for the MCATs. Then you may have to remember the saturation binding curve, and you can study more about that. Fractional saturation depiction Y is a typical, that's the convention for fractional saturation. But please don't worry about it for Bike 410 or Bike 409. All right, so the secret of myoglobin and hemoglobin is in the heme iron. The heme iron position changes the conformation of the polypeptide. It changes it in both myoglobin and in hemoglobin. Without oxygen bound, the heme iron is above the heme plane, out of the heme plane in a dome shape. And then when oxygen binds, it provides the sixth ligand for the heme iron, pulling it from below so that the iron now comes down into the heme plane. And by doing that, the heme, which is coordinated above by that His F8 ligand, the fifth ligand, pulls on the histidine, which of course is part of the F helix, which means that it's going to bring that whole helix down. And so when the helix comes down, when oxygen binds, that actually causes the shift that breaks that salt bridge that I circled on the first slide, and moves those helices with respect to each other in the tetrameric hemoglobin. And that's the nudge that I was talking about that actually causes one subunit to interact to nudge its neighboring subunit and change it from the T state to the R state. Okay, it's a small change, but it means a lot when you have subunits, quaternary structure, hemoglobin. All right, so this restates what I talked about in the last slide, that rupture of the salt bridges between the polypeptide subunits, which could only happen if you have quaternary structure. So that means that cooperativity is afforded for the hemoglobin, but not for the myoglobin. So let's have a look at what that looks like. You can read these lecture notes on your own. Okay, so here now we're looking at the F helix above is in the T state, and the histidine F8 is this blue behind. This blue imidazole ring is the side chain of histidine back here, and the T state of the heme is the dome shaped, right? So the dome shape is behind, and the flattened heme plane, when oxygen is bound, pulls on the heme iron, which is just right here in the middle. And this heme iron, let me write Fe on that. So this is Fe 2 plus, right? And when that oxygen binds to the iron, it as a sixth ligand, it pulls it down, flattening the heme, pulling the fifth ligand, the histidine, down, and the helix that it's a part of, right? Because that's covalently attached, so it's coming down too. So this is the T state up here. This is T up here. And this is R down here. Okay? So that causes a conformational change in myoglobin and hemoglobin, but repositioning the F helix in myoglobin doesn't break any salt bridge, because there was no salt bridge between subunits, because there are no subunits in myoglobin. Hopefully I've hammered that in. All right, so what's happening when we look at the tetramer? There's a 15-degree rotation. So now we're looking at the hemoglobin tetramer from the back. And so this is like a 90-degree rotation from what we saw before. And the front to the front alpha beta dimer is dark pink, and the back alpha beta dimer is light pink in deoxyhemoglobin. Okay, and then as you rotate the front, you're unscrewing the lid. Now you've taken the jar and the lid is facing you, and the glass part of the jar is going into the board. And you twist the lid off by 15 degrees. That's this angle here. So you're not twisting the lid all the way off. You're just twisting it 15 degrees. That's what ruptures those salt bridges that aren't visible here, but they were in that first slide where I circled them in black. Okay, and that allows the oxygen binding sites to be in the R state. So this is oxyhemoglobin, the R state, and this is the T state, deoxyhemoglobin. Their color is opposite from the first picture. I don't know why they did that, but the R state is the oxyhemoglobin, the T state is deoxyhemoglobin, and now you can actually see, or maybe you can see, that you've done this twist that actually causes a conformational change, because these knees, let me do it in red. This knee right here is far away from the knee of the the beta 2. Beta 1 in front's knee, the dark pink knee, is far apart from beta 2's knee in the back. But look, they're touching in the R state, right? You see that? So that's the conformational change that you can see in this view of the deoxyform, or the T state, going to the oxyform, or the R state. So the physiological significance of all this, of course, has to do with their function. That hemoglobin has to bind oxygen in the lungs, which is easy for it to do because the oxygen concentration is so high that it converts the T state to the R state. But then it needs to be able to release the oxygen in the capillaries, something that myoglobin can't do. If hemoglobin behaved like myoglobin, you would get very little oxygen released in the capillaries into the tissues, into the cells, and you wouldn't be able to make ATP, okay? Not very much of it anyway. And so you need that. You need to be able to release the oxygen. And so it's the sigmoidal cooperative oxygen binding of hemoglobin to oxygen that makes its physiological processes possible.